Thank you, everyone, for coming. I am right on time in my practice talk, so I'm going to go right through it. My name is Acer VanWallendael. I am currently a faculty member at NC State, but I'm presenting on some of my research that I did in David Lowry's lab at Michigan State University. But first, a plug for my new lab. It's a little bit different from host-parasite interactions, although we're hoping to collaborate on that. I'm looking at weedy plant evolutionary genetics, both focused on using weeds and invasive species as great models for rapid evolution, and also using evolutionary genetic concepts to better understand weeds. Can the folks in the back hear me okay? All right, no thumbs downs? Okay, great. All right. We are currently looking for a postdoc who has some bioinformatics expertise and is interested in the genetics of rapid plant evolution. So that's my quick plug. Today I'm going to be talking about local adaptation in a, I think, really cool system, which is switchgrass and its leaf fungi. Local adaptation is the process by which species become more well adapted to narrower ranges of habitats. We're really interested in it as a process on its own, but also because it is often a precursor to speciation. The way we test for local adaptation, a couple of folks have done it in many different ways, but the gold standard test is a reciprocal transplant, where we have different regions to which populations are thought to be adapted, and we're going to dig up plants or move organisms to their same region and to a foreign region, and repeat this over space to verify that they actually have higher fitness in their local environment compared to organisms moved there. So this results in a classic cross-reaction norm plot here, where we see that population A does better than population B in environment A, and vice versa. In host parasite systems, we can see multiple levels of local adaptation occurring. We can see adaptation of the host, in this case the plant. You'll see my bias in some of these wordings here. The host to the overall environment, but we can also see local adaptation of the parasite to the host, or the host to the parasite. Usually, parasites have shorter generation times than their hosts, so they can adapt more quickly. So they usually win what can be an evolutionary arms race, but this pattern can be messed up by lower diversity, greater gene flow, or differences in selection. A classic way to test for parasite local adaptation is a version of this reciprocal transplant, where we're going to take parasites, in this case I'm looking at these little butterflies, and move them between host regions. So we have differentiated populations of both the host and the parasite, and we're going to move this parasite around to these different populations and look for fitness advantages between local and foreign transplants. However, there are issues with this approach. When you're moving things like butterflies, people don't tend to get too mad, but aphis will knock down your door if you start moving pathogens around, especially if they're for agricultural crops. So one variant of this particular type of experiment is to, rather than transplanting the parasite, we're going to move the host itself into different parasite populations. And what this would look like in the toy example I have here is taking this mustard plant and moving it into the northern population of the white butterflies and also the southern population, and then moving the southern host as well to both regions. We can actually test using the same way, but it's kind of a flip in the mind of how we think about what the environment is. Rather than thinking of the global environment of an organism, we're thinking of the host as the parasite's environment. So we can move the environment, in this case, which solves some of the issues ethically, legally, and just kind of experimentally with moving the parasite around. And often you can actually do this in the field rather than a lot of parasite transplants, which need to happen in controlled conditions. This approach has been used a couple of times, actually often in other plant rust host parasite systems, but no one has given it a name, so we just went with post-reciprocal transplant. If anyone hates it, I apologize. So the system I'm working in where we tried this is switchgrass. Switchgrass is a large prairie perennial grass, and it has been studied for a while in a evolution and sort of breeding genetics context, because it does have these widely different ecotypes adapted to different regions of North America. The classic examples are this lowland ecotype, which often comprises the Gulf population, genetic population, and the upland ecotype, which is considered part of the Midwest ecotype and is more adapted to northern regions. These often have differences in their tolerance to fungal pathogens. So we focused on one particular fungal pathogen called leaf rust, and we asked a couple questions. First, to do this host reciprocal transplant, we had to know that these leaf rusts were actually different across regions that we were transplanting to. Otherwise, we would just be kind of moving these hosts to undifferentiated populations. We wouldn't see the signal that we're hoping for. So we had to check that first, and then we did our local adaptation test. And then finally, we know a lot about the genetics of these plants, so we're able to actually look through the genome for particular genetic loci that explain rust severity across these populations. So this is the study design we have here. This is a huge collaboration with between folks at MSU, Missouri, and University of Texas particularly, and a couple other folks who manage field sites. These plots are huge. We take, it might be tough to see here, but all the little dots on this map are collections we sampled switchgrass genotypes from. And then the X's are the common garden locations throughout the regions that we transplanted them into. And the larger gardens look something like this, where we have many different switchgrass plants planted together. We looked at rust populations across states. So this is a plot going from north to south and looking at the first principal component of the genetics of the switchgrass rust pathogen. And we see that there are largely different genetic groups that correlate well with latitudinal differences. And these are pretty well separated into sort of a northern population and a southern population. So fortunately, we did see support for the idea that these are actually genetically differentiated strains, varieties, something of this rust. And these correspond well with particular switchgrass populations. So the northern rust corresponds well to the general region of Midwest switchgrass, the southern rust to Gulf switchgrass. And we didn't actually have any of our common gardens in the core of the range of Atlantic switchgrass. So we don't know what the rust, or we didn't know what the rust was going to do on it. We measured all 10 of these sites over 3 years, a bunch of genotypes, and multiple sampling time points. And all that got compressed down into one tidy box plot graph. A lot of work. So we looked at area under the disease progression curve, which incorporates information from multiple time points. And then we also kind of compiled our information across sites and years using this mixed linear model, which allows us to estimate the best linear unbiased predictor of the random effect genotype, which is a long way of saying the y-axis corresponds to the susceptibility of each genotype to rust, or thinking about it the other way, the fitness of rust on each particular genotype. So in the Atlantic population, I keep thinking I have a pointer there, but the Atlantic population did not show any differences across the regions we grew it in. So the rust was equally unsuccessful in both regions on the Atlantic population, which makes sense since they're not co-evolved together. We did see the pattern that we expected in the case of local adaptation. where in the Gulf population, the southern Gulf area rusts were more successful on those switchgrass coasts. And in the north, the Midwest population was more susceptible to northern rust. So these rusts are locally adapted. And that's what we expected based on the shorter life cycle. But it is a little bit surprising, since normally when we think of host-parasite local adaptation, we think that if the pathogen is locally adapted to the host, then the host will be maladapted, or will not show up having a local adaptation. Switchgrass is a classic example of a locally adapted organism, especially across this region. So how can we explain this discrepancy? This is a special case where we see that what I believe is happening here is that the abiotic selection on switchgrass is a good bit stronger than the selection imposed by the pathogen. Unlike in Sam's talk earlier, these are not causing complete fitness decrease in the plants, whereas we do see a really strong selection from the abiotic environment, particularly winter kill in these populations. But the pathogens will come back year after year, and you'll see some decrease in biomass from them, but they don't totally kill the plants. And this has been seen in a couple other systems as well. So the last question I wanted to look at was on the genetic basis of this interaction. So we had a previous paper using quantitative trait locus mapping to look at the different regions in the genome that are associated with rust severity. We know from Sam Yeaman's talk yesterday that local adaptation often involves relatively few large effect loci across the genome. And we actually do see that pattern in the QTL. So we had 2 large effects QTL, especially when we grew these plants on northern pathogens. However, this was only 4 genotypes. So we wanted to look across our GWAS panel. We do actually see a very different pattern in the GWAS panel. We see a ton of loci associated with variation in rust severity. Although we do recapitulate both the 3N and the 9N QTL, there are many other regions of the genome that are associated with variation in rust. And one of the reasons I think this is happening is we're not actually observing local adaptation of switchgrass. Switchgrass is not locally adapted to the rust, so we don't expect to see those few large effect loci. We also see some indication that this is not the classic R gene mediated resistance. In this case, we might be seeing metabolite mediated resistance, which is often more polygenic. There's a lot more in this paper. It's a really fun one. We just published it on bioRxiv. The fastest way to find it is to go to bioRxiv and search Acer's on race, which is also a podcast I'm releasing soon about my weightlifting journey. Thank you all for coming, and thank you to everyone who contributed to this.
All right. Hello, everybody. My name's Emlyn Rasetarits. I'm a new assistant. Okay. Hello, everybody. I'm a new assistant professor at Barnard College at Columbia University, and I'm going to be talking to you today about collating a bunch of data that people have been collecting over the past 20 years to look at an inverse latitudinal gradient in this wide-ranging snail species that I study. So the species that I study is the eastern mud snail, Ilionassa obsoleta. It's a really abundant grazer on the East Coast. You can find it from Florida all the way to Maine. It's also become an invasive species on the West Coast, so there's a lot of problems with it on mudflats on the West Coast. But it's super, super abundant. We see it in mudflats and sandflats, and you can see these densities that we get. We can get up to about 2,000 snails per meter squared. So they're really important for these estuarine ecosystems, and they're also really easy to collect, which is great. Now, the reason why I really like the species is that there's a diverse suite of trematode species that use Ilionassa obsoleta as their first intermediate host. So here I'm just showing 6 ones that we find most commonly in our snails. And what I'm actually showing here are the free-swimming stage of this parasite called Cercaria. And by looking at the Cercaria under the dissecting scope, usually what we do is we look at them under a dissecting scope, we crack them with a hammer, and then it's very easy, these Cercaria pour all out, and it's very easy to identify which of the 9 species is infecting the snail. So as I just mentioned, there are 9 species that use Ilionassa obsoleta as its first intermediate host. And if you're not familiar with trematode life cycles, I'll tell you a little bit about them. So they have these complex life cycles, which make them really ecologically interesting. So first, generally, the definitive host where sexual reproduction happens is in either birds, fish, or the diamondback terrapin in this case. And sexual reproduction happens there. These hosts defecate eggs out into the water column, which then get ingested by the eastern mud snail. These eggs then form a colony in the gonads of the snail, thus castrating these snails. Generally, then that cercarial stage that I showed, that free-swimming stage, will leave the snail and then insist on a secondary intermediate host that really varies widely between trematode species. So they can be invertebrates, bivalves, fish, polychaetes, things like that. And then usually to get to that definitive host again, they need to be trophically transmitted, i.e. they need to be eaten by that final host. And so what I'm showing here is I've kind of grouped them. So the top ones are all the ones that use birds as definitive hosts, then the one that uses the terrapin, and on the bottom, all the ones that use fish. So this project really emerged kind of by accident as due to some fieldwork failure. So my previous position was a scientist in residence at the UGA Marine Institute, and my original goal for the project that I was trying to do out there was to collect a bunch of infected and uninfected snails, because we can non-invasively determine infection status, and then look at their behavior and see if they have different ecological roles on these mudflats. That was the plan. What it ended up being was just me desperately trying to find enough infected snails across Georgia to even do these experiments, which was really frustrating and actually kind of confusing to me because my postdoc advisor about 20 years ago had done a very diverse survey across Georgia and had found maybe not high, but definitely higher parasitism than what I was finding. So the first thing that I did was trying to see, okay, am I actually seeing lower parasitism in Georgia than my advisor found 20 years ago? So yes, there does seem to be about a 5% decline. Well, I guess about a 30% decline, right? So it's going from on average 15% of the snails are infected in Georgia to now about 10%. And this is significant. So in an attempt to turn lemons into lemonade, this set me on a mission to try to work with all of the people who I knew had collected data on Ilionassa obsoleta and its parasites to try to get, to collate this data and see what we might find across its entire range. So before I went into this, we did know a couple things about Ilionassa across its entire range. So one thing we know from previous work by April Blakesley and colleagues is that northern populations on average have higher prevalence and richness. So the first figure is showing species richness in the north has significantly higher than the south, and then the second figure is showing prevalence, and you see again, on average, higher parasitism in the north than the south. And this is actually pretty strange in terms of diversity, because usually what we see is a diversity gradient where as you go towards the tropics, in free-living species, you see higher species diversity, but that's not what we see in our system based on this preliminary work before. And this is not the first instance of trematodes having a different latitudinal gradient of richness. So previous work that I'd done during my PhD, along with Mark Torchin at Stry and his colleagues, was to demonstrate that another wide-ranging marine species also had gradients of infection prevalence and diversity. So this one's just showing Cerathidia california, or the California horn snail, and it actually has an interesting latitudinal gradient where it has highest prevalence in the intermediate area of its range around San Diego, Northern California, and then as you go to the north, parasitism, richness, and prevalence goes down, as well as when you go towards the tropics. We also know that complex life cycle parasites, such as trematodes, are really excellent for tracking free-living host diversity and abundance. So multiple studies have shown that parasite richness is higher in restored or pristine habitats. So on the, I always get my left and right confused. On the right figure, you can see Moore et al. Tracked fish parasite richness after a restoration efforts, and you can see that parasite richness increases years after a restoration. And then Wood and Lafferty also compared fished and unfished atolls and found that unfished atolls also had higher parasite richness of these complex life cycle parasites. So I wanted to collate as much data as I could, and there was 4 main questions that I wanted to ask with this data. First, how does trematode infection vary across latitude? Has trematode infection changed over the past 20 years? Is it increasing? Is it decreasing? And do these results vary between these different trematode species or between different groups of definitive hosts? Do the bird trematodes differ than the fish trematodes, for example? And then what can this tell us about free-living diversity? So for this project, I compiled published and unpublished data from this amazing group of Illionassa obsoleta researchers who have been collecting prevalence data across the East Coast for the past 20 years. And so far, I've collected, we've got about 15,000 snails, so we have individual infection status and information on individual snails for these 15,000 snails across 110 sites along the East Coast over the past 20 years, spanning 14 degrees of latitude and 13 states. And so this is a map of all of our sites. So you can see we've got a really nice coverage across the entire East Coast, and we also have nice coverage over the past 20 years. Additionally, I also added some additional climate data for each of our sites so that we have, I took data from WorldClim, which has data from 1970 to 2000, and looked at average temperature, average minimum yearly temperature, and average maximum yearly temperature to see if that might explain some of this data more so than latitude. So comparing latitude and these other environmental variables that we know vary with latitude. And this data represents historic data that's summed across those 30 years, not temperature data for the year that the snail data was collected. We also estimated the number of months below freezing, since the growing season can strongly impact snail life history traits. We didn't find this to be significant for any of our models, so I'm not going to talk about it anymore, but we did look at it. And then I ran model selection for overall prevalence, richness, and individual trematode species prevalence. And the model variables that we used were year, climate variable, latitude, max temp, or months below freezing, and then whether or not there was an interaction between year and that climate variable. Okay, so what's going on? What do we find? So at the bottom, you can see this table. Sorry to put a table in here, but just to orient you to the 10 different variables we were looking at and what explanatory variables came out in our best fit models for each. So we've got overall prevalence, which is summing all of the prevalences for our different trematodes. So what percent of the snails were infected with any trematode, richness of trematodes, and then our individual species, trematode species prevalence. So the first thing that I want to show you is that 4 of our trematode species show significant effects of latitude. So AV and DN are both bird parasites. PM is the terrapin parasite, and then Stephanostomum species are a fish parasite. And all of them show that in northern latitudes, you have higher prevalence on average. This isn't terribly unexpected, since we knew that in the north we find higher parasitism on average. We did find that maximum temperature better than latitude explained a couple of these variables. So both richness and HQ prevalence, which is a bird trematode, show that they go down at average maximum temperatures, right? So in northern areas that are on average cooler, we have higher richness and higher HQ prevalence. I think the most interesting thing that we found, and wasn't actually what I necessarily set out to look at, was that we saw significant declines in infection prevalence in 4 of our species, all of the ectotherm species. We also find that total prevalence and richness decline over the past 20 years. And to just put this into context, if you look at total prevalence, in general, what you see is that the decline is about 50%. So some other interesting things that I'll just really briefly touch on is that we did find for 2 species an interaction between year and latitude. So Pleurogonus, which is the terrapin parasite, it seems to be declining over time in the south and increasing over time in the north. And then Stephanostomum is showing stronger declines in the north than it is in the south. So to summarize what I found, we found that bird, terrapin, and fish trematodes are all increasing with latitude. We found that richness and HQ prevalence, which is one of those bird parasites, has declined at warmer maximum temperatures. And most interesting to me, and what we're going to be looking at in the future in our lab, is that overall prevalence and richness have decreased over the past 20 years, particularly these ectotherm parasites, or species that use ectotherms as their definitive hosts. So I'll just leave you with some implications of this. So fish and terrapin parasites appear to be declining over time, suggesting that these ectotherm hosts may be declining in these areas as well. And then I'll just leave you with one quote. If parasites are sensitive to host species losses, then ironically, their presence might indicate rich and functioning ecosystems. Low parasite richness suggests a system in trouble. In other words, do not wait for the canary to die. You should leave the coal mine when the canary loses its parasites. Thank you.
Yay. All right, I'm going to breeze through a lot of the introduction so we can get to the data. So my name's McCall Calvert. I'm a PhD student in Corlett Wood's lab at the University of Pennsylvania. Really excited today to talk to you about some work we've been doing on the fingerprints of host-associated selection and the genome of a hypergeneralist parasite. So I'm going to breeze through some of these. Basically, there's a lot of specialists out there, right? A lot of specialist parasites. There are, of course, generalists as well. They represent a smaller proportion of biodiversity. But due to the sort of ubiquity of more specialized interactions, as well as their experimental and theoretical tractability, a lot of what we've learned about the evolution of parasitism comes from those systems. And by comparison, we know less about how generalists evolve, and in particular, we know less about how heterogeneous host selection shapes variation in those generalists, which is something I'm really interested in. And that brings up kind of the main objective of this project, which is thinking about what patterns does heterogeneous host selection leave in the genomes of generalist parasites. And so this question, is not totally unaddressed. People have thought about it, but a lot of work in this area has actually been done on parasites, on taxa that maybe appear to be generalist at the level of the species, but are actually comprised of fairly diverged and fairly specialized populations. An example of this comes from Rhagylides palmonella, or the apple maggot-fly, which is not a parasite, but still an antagonist, where at the species level, they infest a variety of different species of host tree, But this species is actually comprised of very specialized populations that really only infest one tree, or one tree species. By comparison, we know a lot less about how selection operates in the genomes of what I would describe as hypergeneralists. So these are generalist species that have tens to hundreds of known taxa, and there's not much evidence of genetic differentiation, at least host-associated differentiation across those taxa. And some examples of these would include things like different species of Leishmania parasites, which infect mammals, chytrid fungus, which is a devastating pathogen of amphibians, and the focus of my talk today, which will be these root knot nematode parasites. And so some of the main questions we were interested in asking with this project was, are there genetic loci that are associated with host selection? And if there are, do they overlap across different hosts? What form of selection is operating on these loci, and what can that tell us about the evolutionary process in generalists? And then finally, are host loci enriched for specific functions, and can that tell us something about the generalist phenotype? And we did this work in the northern root knot nematode. It gets its name from these kind of nasty-looking gall structures, or knot root knots. They begin their life as a migratory juvenile. They'll move through the soil looking for a susceptible host plant. When they find one, they will invade the root system. They will they'll develop a feeding site. They'll molt into a sedentary adult where they'll reproduce and eat for the rest of their life. And the species that we work on in particular is Meloidogyne hapla. And so here's just a distribution of the number of known host plant families that different nematode parasites infect. And you can see most of them seem to be fairly specialized. Meloidogyne hapla is out there in the tail with over 80 known host plant families. This comprises hundreds of host plant species. So we sampled root knot nematodes from 3 sites in Virginia, 1 in Pennsylvania. And at each of these sites, we sampled nematodes from 3 different host plant species, 1 belonging to Fabaceae, 2 to Asteraceae. You can see here the numbers of nematodes that we got from individual plants belonging to each of these species. I'll be mostly focusing entirely actually today on the Virginia samples, but the Pennsylvania samples are cool too. Something that we're really excited about is the workflow that we use to generate data for this project. So traditionally, there's been several barriers to sequencing, very tiny parasites from the field. But we applied whole genome amplification to this project, which is a technology that was really optimized for whole genome sequencing, for single cell sequencing. But it allowed us to get whole genome libraries from individual parasites in the field, very tiny, barely visible parasites. And so we got these individual libraries. We aligned reads to the haploreference assembly from Wormbase. And after standard variant calling and filtering, we had 117,000 SNPs to work with. And before jumping into the main results of this project, I just want to show you the results of a PCA with some putatively neutral, unlinked SNPs that really highlights that there's not really any evidence for cryptic host specialization in this species. So in this plot, each point is a different individual nematode. They're colored by what sampling site we collected them from, and then the different shapes indicate the different hosts. And you can see very clear geographic structuring and not really any host structuring. And so what this indicates is there's not really strong enough host-associated selection or strong enough barriers to gene flow to generate very clear host-associated ecotypes. So on to our first question, which is just whether the same genetic loci are responding to selection from these different host species. We did this by performing a version of a genome environment association analysis in BayPass. But basically what this allows us to do is compare allele frequency differences between nematodes collected from different host plant species while controlling for the underlying structure that exists across our sampling sites. And the results of this just look like, you can plot them like a Manhattan plot. You can see how fragmented our reference genome is. But just the only thing to pay attention to here is that on the y-axis we have this contrast statistic, which is a standardized allele frequency difference, standardized and scaled, which is why it goes above 1. But you can see that there's lots of hits that we get. And this is true across the other comparisons. And there's also a fair bit of overlap, a sort of non-random amount of overlap across these different genomic regions. We were also curious about what form of selection could be operating on these host-associated loci. In the interest of time, because I had to start late, we'll skip some of the logic here, and I'll just jump straight to the results. So we assessed patterns of selection using Tajima's D. The only thing really to know here is that low values of the statistic indicate an excess of low-frequency polymorphisms, often taken as evidence of positive selection. And high values indicate excess of intermediate frequency polymorphisms or signals of balancing selection. And we used coalescence simulations from a best-fitting demographic model that allowed us to put statistical thresholds on this to decide whether a specific genomic region was, gave us evidence of selection. And so really, these are just going to be enrichment plots. In the interest of time, I'm just going to breeze through them. The main thing to take away from these is that we see enrichment of genomic windows that exhibited signals of positive selection were enriched for SNPs that were host-associated, right? And we see this across all of our populations, but also within each of them. And we see a very similar signal for balancing selection. What's interesting, too, here is that there's a similar frequency of signals of both positive and balancing selection in our data set. So it's not like one or the other seems to be happening more. And just to interpret this, and this is the table that I had in the previous slide that I breezed through, is just that depending on whether you kind of think that this generalist parasite is actually engaged in some kind of ongoing coevolutionary interaction with its hosts, maybe diffuse coevolution, or if it's maybe sort of unidirectional local adaptation, the way that you would interpret whether positive or balancing selection is happening, or what the meaning of those two different types of selection kind of indicate, different things, which I've listed here. And I'm going to breeze through this. We see host-associated SNPs enriched in introns. Something that's really, really cool is there was some previous work done looking at cross-species EQTL hotspots. These are just regions of the parasite genome that influence host genetic gene expression, variation in gene expression. What's really, really cool here, and I'm breezing through this, so I'm sorry, and I can talk to you guys after this if you're more interested. What's cool is that we see strong enrichment of signals of balancing selection in these EQTL hotspots, so they're hyperdiverse, as well as a strong enrichment of host-associated SNPs in these regions, which further implicates the functional role of the variants that we saw. And so just to conclude, we saw a lot of really cool host-associated variation despite the lack of evidence of host-associated ecotypes, very clear signals of selection that we're really, really excited to dig into more to think about whether this is indicative of resulting from ongoing coevolution, and then clear patterns of, sort of evidence that there's some functional role for this variation. Just want to give a huge thanks to my lab mates, and if you have any questions, just talk to me afterwards. Thank you.
Okay, well, thank you everybody for making the time to come out to my talk. I'll be telling you all a little bit about the work I've done so far on my project looking at schistosomiasis. So schistosomiasis is a neglected tropical disease caused by worms in the genus Schistosoma, which is a helminth. It's found mainly in regions of sub-Saharan Africa and Asia and affects over 200 million people on an annual basis, with fatalities ranging in the thousands every year. It's considered second only to malaria regarding socioeconomic impacts in developing countries, as its effects are known to be pervasive and poverty reinforcing. For this project specifically, I'll be looking at Schistosoma japonicum, which is located in Asia. Specifically, I'll be looking at Schistosoma japonicum in China. There's been a long history of schistosomiasis in China. In the 1950s, more than 10 million infections were estimated to be spread across 12 provinces. However, through a rigorous, multi-pronged control program that lasted over 70 years, which included the development of better sanitation practices, molluscicides, and mass drug administration of the anti-helminth drug praziquantel, cases have been reduced drastically. As of 2013, there were reported to be less than 200,000 cases spread across 5 provinces. However, as cases are disappearing across China, located here in Sichuan, there has been continual reemergence of this disease noted since 2006, despite strong control pressure within this region. A question we really want to figure out is why is schistosomiasis still prevailing despite these intense control measures? And we plan to look at how these intense control strategies may have affected life history traits in Sichuan and how potential selective sweeps have contributed to reemergence. Really quickly, Schistosoma japonicum has a very complex multi-host life cycle. Eggs are excreted from a mammalian host into a water column where they hatch into their meracidia or larval form. They then penetrate an intermediate snail host and undergo asexual reproduction or cloning. And then the free-swimming Saccharii will penetrate back into a mammalian host where they move into the liver and grow into their adult forms, where they then sexually reproduce, secreting eggs back into the environment. So we used around, did I mess up? We will use around 500 individuals sampled from 34 different villages between 2007 and 2019. So this timing actually coincides with the transmission outbreak that's still ongoing in Sichuan that was noted in 2006. And we targeted multiple Myristidia or larvae per host to maximize the overall diversity of our sampling. Like my previous presenter, we also used whole genome amplification. Because the sequencing of individual Myristidia is very tough, as they only have 1 to 2 nanograms of DNA, the whole genome sequencing allowed us to get whole genome, the whole genome amplification allowed us to get whole genome sequencing from archival samples that can be up to 20 years old. And we now have over 500 whole genomes of Schistosoma japonicum. To answer why we think Schistosoma japonicum is still undergoing reemergence, we used rare allele sharing across individuals to better understand relatedness between samples and hopefully uncover some of their transmission dynamics. This figure here is a heat map of rare allele sharing between all sampled Schistosoma japonicum Myristidia from 2007. So it's a mirror image showing the relationships between different hosts and villages. And here we see patterns of transmission events being localized within villages, where we see high levels of relatedness among a group, and then lower order relationships being more common between the different villages. So this is letting us know that despite multiple rounds of mass drug administration on a local and regional scale, there are still local level infections persisting in villages, and these drugs are not effectively eliminating source reservoirs. A potential problem for drug efficiency is that there may be multiple host reservoirs. We have sequenced 13 bovine samples to try to explore the role secondary hosts play in transmission dynamics. This is a preliminary PCA of our whole genome data of both human and bovine samples, and you can see that the bovine samples cluster really neatly within the human samples. We think this may suggest that parasite populations may be a single large populations instead of 2 distinct populations separated by host. We predict that the strong control measures such as mass drug administration have reduced prevalence of schistosomiasis so much in humans, but have had likely little impact on bovines, which may represent them being a significant reservoir, which consists to regional persistent human infections. So to further test for selective pressures, which could respond to strong control strategies, I used publicly available genomes by Liu et al. For pairwise comparisons to our Sichuan Inc. group. The countries we choose to draw from are the Philippines, Indonesia, and Taiwan. There is a huge contrast in control measures between these different countries, as well as different environmental challenges to control. And I will use tests such as XPEHH and FST to test for evidence of the selective sweeps that would be unique to Sichuan. First, to understand if there's evidence of selection, we use XPEHH, or a cross-population extended haplotype homozygosity, which is a statistical method used in population genomics to detect genomic regions that have undergone recent positive selection by looking for a significant excess of a long run of homozygosity within 100 kb windows in a particular population compared to another population. So here are multiple pairwise comparisons between Sichuan and other countries. We have Sichuan Indonesia, the Philippines, and Taiwan. So this plot is a whole genome scale using the XPHH, where the solid black lines are representing the average score within a 100 kb window, and red dots are indicative of windows that had p-values in the top 5% and were shown as showing evidence of a selective sweep. As you can see, this test gave us many different regions across a whole genome that we can look at for multiple comparisons. However, we wanted to focus and reduce by merging what was unique to Sichuan across all of these comparisons. So once we got our polarity scores provided from the XPEHH test, we further reduced by seeing the regions that were unique only to Sichuan across all 3 comparisons. So we eventually reduced down to 7 regions, which are shown to have experienced recent positive selection across the whole genome. To take a further look at this, we looked at FST for each of these regions. Here's an example from chromosome 3. So this is genomic scans of FST and chromosome 3 in all pairwise comparisons. The lines are showing FST in 10 kb windows, and the dots are 1 kb windows. And you can actually see this is really interesting because that dark blue line is annotating an annotated gene, and you can see that FST is very high across all 3 comparisons, which could be showing evidence for distinct allele differentiation and perhaps unique and fixed variants within this population. So we decided to take a look more into the genes within these regions and found that across the 7 genomic regions identified by having potential strong selective sweeps by XPEHH, there was a total of 30 genes, 21 of which have functional annotations. This graph is showing FST scan of all outgroup populations against Sichuan in 10 kb and 1 kb windows on 3 of the 7 identified regions consecutively on chromosome 3, 4, and 5. So we thought it was really interesting that through this just naive whole genome scan approach, we identified multiple biologically relevant genes that are associated with drug resistance. For example, we had DHFR on chromosome 3, which is a critical enzyme for DNA replication and has been considered a therapeutic target for multiple drug treatments due to previous research showing it as a potential mechanism for drug resistance. We also have the nicotinic acetylcholine receptor alpha 1, which is a part of a sodium ion channel reception and is closely followed by TMEM 168 and RPE, which are transmembrane proteins also linked to drug resistance traits. Finally, we have ABCB7 on chromosome 5, which is a member of a large family of ATP-binding ABC multidrug transporters. This drug is shown to be important to biological functions within the schistosome and is also associated with multi-drug resistance. All of these potentially relevant genes have made us want to take a closer look at the biological variants. So we used SNP-EFF program to predict the high-effect SNPs within these regions. We found evidence of a number of moderate impact variants and interestingly saw that the 3 consecutive genes shown on chromosome 4 all have non-synonymous mutations within them. So far, as we begin this analysis of this project, we just thought it was really remarkable that these naive genome-wide scans seem to clearly point to drug-resistant genes in a population undergoing some of the strongest mass drug administration program in the world. Overall conclusions from this project so far is that it appears that strong control measures have failed to eliminate local sources of infections within villages, and that alternative mammalian hosts, such as bovines, may complicate population dynamics by acting as source reservoirs. And there is preliminary evidence that strong control measures can lead to rapid adaptation in parasites, including drug resistance. So overall, this project really highlights the value of genomic surveillance to keep pace with pathogen evolution and to better understand elimination dynamics. Obviously, in a post-COVID world, we all understand how important genomic surveillance of pathogens can be and to better understanding how to eliminate them. Some next steps that I plan to take for this project include expanding analysis to better understand the origins of selected alleles by increasing our temporal and geographic sampling, as well as get a detailed functional characterization for selected alleles to look for potential biological relevance. So now that you know a bit more in depth about this region of China and the problems concerning there, this is a reminder of why this area is so important. So China has probably the most advanced stage of schistosome control and elimination worldwide, but as hotspots are actively emerging in parts of Africa where control programs rely heavily on mass drug administration and public health officials are trying to expand control strategies, our study region provides a very important case study on how to better assist with elimination dynamics within those regions in Africa. And with that, I'd like to thank my awesome lab mates, my collaborators, and my funding sources at the NSF and the NIH, and my PI, Dr. Casto, and you all for your time and attention, and I'll take any questions. Oh, yeah.
This might be my own ignorance of the system, but is it possible that with this bovine potential reservoir, the genes for resistance could be coming from this other environment? And mutations arising within?
Yeah, so I think that's really interesting. We actually have identified in schistosomes the gene that codes for host preference. So we're actually going to begin to look at, see if any selection has been noticed in these genes that mark for host preference, to see if maybe they're switching more to bovines as opposed to humans, which could